This is Biology for Grade 11.
Hello students, today we will see on Unit 4 Cell Biology. Here we have two subtopics. The first is types of cell and the second subtopic is part of cell and their function. From this lesson, the students are expected to know what prokaryotic and eukaryotic cells are, to mention the main difference between prokaryotic and eukaryotic cells, to explain the key features of fluid mosaic model. Before we start our today's session, let's recall some points from your previous lessons, especially about cells. The word cell was first coined by Robert Hooke in 1665 from a dead cord plant tissue. Based on the modern cell theory, cells are the basic structural and functionality of life. Cells comes from pre-existing cells, not by spontaneous generation. Cells contain hereditary information that passes from generation to generation or from cell to cell during cell division. All metabolic activities occurs within the cell. All organisms or living things, they are composed of one or more than one cells. If those organisms are composed of one cell, we call them unicellular organisms. For example, like bacteria, yeast cell, protozoan cells, and others. Whereas those organisms that are composed of more than one cells, they are named as multicellular organisms. For example, like plant cells, animal cells, funguses like mushrooms are multicellular organisms. Having this in mind, let's proceed towards our today's main session, that is the first subtopic, types of cell. Based on whether the cell has true nucleus or not, there are two types of cells, prokaryotic cells and eukaryotic cells. Students, what do you remember about prokaryotic and eukaryotic cells from your lower grades? Okay, students, let's see the main features and differences between the two types of cells. Both prokaryotic and eukaryotic cells words are derived from Greek. Pro means before and karyos means nuclear. So prokaryotic cells are cells that have no distinct nucleus or cells that have no true nucleus. Whereas EU means true, so eukaryotic cells are cells that have a true nucleus or distinct nucleus. Prokaryotic cells are much smaller and simpler cells. They ranges from 1 up to 10 micrometer in size, whereas eukaryotic cells are large and much complex cells. They ranges from 10 up to 100 micrometer in size. Prokaryotic cells, they have no membrane-bounded organelles, whereas eukaryotic cells have membrane-bounded organelles like nucleus, mitochondria, chloroplast, lysosome, Golgi apparatus, and others. Prokaryotic cells have a circular DNA that is not associated with histone protein to form chromosome. However, eukaryotic cells have linear DNA associated with histone protein to form a chromosome. In addition, prokaryotic cells have small ribosome, that is 70S. S stands for sedimentation coefficient that measures the mass of the ribosome. Prokaryotic cells, they have always cell wall made up from peptide glycan. Some prokaryotic cells, they are photosynthetic. For example, cyanobacteria or blue-green algae. Cyanobacteria or blue-green algae, they have phycocyanin pigment instead of chlorophyll pigment. However, eukaryotic cells, they have large ribosome that is 80S. Some eukaryotic cells have a cell wall. For example, plant cells have a cell wall that is composed of cellulose. Fungi cells have a cell wall that is made up from chitin. And algal cells have a cell wall that is mainly composed of glycoprotein. Plants and algae are photosynthetic organisms that have a eukaryotic cell type. Many biologists, they believe that prokaryotic organisms are the first cells that evolved on the earth. Based on the evolutionary point of view, or specifically based on the theory of biochemical origin of life on the earth, The first true cell that derived from coacervates are named as protobionates. Protobionates are prokaryotic, heterotrophic, unicellular organisms. Prokaryotic cells, they include this general bacteria like Archaeobacteria, Cyanobacteria, and Eubacteria. Let's see one by one. Archaeobacteria are the primitive prokaryotics and the oldest organisms on the earth. Cyanobacteria are the first photosynthetic organisms evolved in the earth around 3.5 billion years ago. Eubacteria are the ordinary bacteria such as Shigella, Salmonella, Campylobacter, Escherichia coli, and so on. Eukaryotic cells includes all organisms in kingdom Fungia, kingdom Protista, kingdom Plantae, and kingdom Animalia. Indocyanebionate theory. Indocyanebionate theory is the theory that states about how eukaryotic cells originated. So, based on this theory, the modern eukaryotic cells formed when several primitive prokaryotic cells got together. The plasma membrane of those prokaryotic cells that got together becomes more enfolded and eventually leads into the endoplasmic reticulum of eukaryotic cells. The membranous cell that has endoplasmic reticulum engulfed other smaller cells that are better in respiration to release energy, and they evolved into mitochondria. Those cells that contain mitochondria were heterotrophic and evolved into Protista, fungi, and animal cells. Some of the cells that have mitochondria, they engulfed other smaller prokaryotic photosynthetic cells, and they evolved into chloroplast. They are autotrophs, and they are the forerunners of plant cell and algal cells. Students, are you with me? If you are with me, let's try to answer the following questions. Question number 1. What are the two components of a eukaryotic chromosome? What are the two components of a eukaryotic chromosome? Okay, students. If you say DNA and histone protein, you are right. Question number 2. What are phycocyanin pigments? What are phycocyanin pigments? Okay, students. Phycocyanin pigments are blue-green colored pigments that are found in cyanobacteria that help us to capture sunlight that use it for photosynthesis. Question number 3. Mention at least two differences between prokaryotic and eukaryotic cells. I will repeat the question. Mention at least two differences between prokaryotic and eukaryotic cells. Okay, students. Prokaryotic and eukaryotic cells have a number of differences. If you say two from the number or from the several differences, you are right. So the differences are prokaryotic cells, they have no true nucleus, whereas eukaryotic cells have a true nucleus. Other difference is prokaryotic cells, they have no membrane-bounded organelles, whereas eukaryotic cells have a membrane-bounded organelles like mitochondria. The DNA of prokaryotic cells is circular, that does not associate with histone protein to form a chromosome, whereas the DNA of eukaryotic cells is linear, that associated with histone protein to form a chromosome and others. Okay, students, here you have homework. There are 5 multiple choice questions on page 124 up to 125 of your textbook. write them on your exercise book properly. Do them for next session. Okay, students, let's proceed towards our next subtopic, that is part of cell and their function. As we know, cells have different parts. Today, we will mainly focus on cell membrane or plasma membrane. Cell membrane or plasma membrane is a cellular structure that surrounds and encloses the cell, and it is vital in isolating the cell from its environment. Cell membrane can be described as selectively permeable membrane, or differentially permeable membrane, or partially permeable membrane, or semi-permeable membrane. Plasma membrane or cell membrane controls what enters and leaves from the cell. Because of this, it is the traffic of the cell. Plasma membrane uses as cell signaling, or it allows the cell to recognize and identify various types such as hormones, immune system, etc. Plasma membrane also gives a little mechanical support to the cell. When we come to its composition, plasma membrane is mainly composed of a phospholipid bilayer. There are different models that shows the structure of plasma membrane. Hereunder, we will see 3 models. The first one is sandwich model. This sandwich model was proposed in 1935 by Davizene and Danieli. It says a phospholipid bilayer is sandwiched in between 2 protein molecules. That means the protein forms the bread of the sandwich, whereas the phospholipid forms the filling or the center of the sandwich. However, this sandwich model does not properly explain how molecules passes across the membrane. The second model is unit membrane model. This unit membrane model was proposed by Robertson in 1959, based on electron microscope evidences to support sandwich model or Davizene-Daniel model. This unit membrane model suggests all membranes are essentially the same in their composition. The third model, that is the acceptable model, the model that we use at this time to represent the structure of plasma membrane is fluid mosaic model. Fluid mosaic model was introduced by Singer and Nicholson in 1972. It is the acceptable model as we said before. It states that the membrane is a mosaic of proteins dispersed in the fluid bilayer of phospholipids. That means the presence of different kinds of proteins and others shows mosaic. And the phospholipid is not static by its nature. It is fluid and it constantly changing. Fluid mosaic model has the following key features. The first one is, it says phospholipid bilayer is the basis of the membrane. The second key feature is plasma membrane has integral proteins. Integral proteins are also called intrinsic and transmembrane proteins. Those integral proteins span or lies on both phospholipid bilayer. They play a role in transporting substance. Those integral proteins, they consist of 3 major groups of protein that are channel protein, carrier protein, and peripheral proteins. Let's see one by one. Channel proteins are integral proteins through which specific ions can pass. Different ions have different channel protein. The second protein is carrier protein. Carrier proteins are integral proteins through which large molecules cross the membrane by facilitated diffusion or active transport. Those carrier proteins that involved in active transports are named as pumps. The third integral protein is peripheral protein. They are also called extrinsic proteins. Those peripheral proteins span or lies only in one layer or less than one layer of the phospholipid. Those peripheral proteins, they act as enzyme and catalyze a reaction. Other peripheral proteins, they attach the integral protein to cytoskeleton of the cell. Okay, students, let's continue with the other key features of the fluid mosaic model. That is the third one, presence of glycolipid and glycoproteins in the membrane. Glycoprotein and glycolipids are proteins and lipid molecules that attached with carbohydrate chain. Both glycoprotein and glycolipids, they act as a receptor sites for hormones and drugs. Also, they allow cell-to-cell recognition, mainly by their carbohydrate part. Glycoproteins, they also works in sticking the correct cells together in tissue. The fourth key feature of fluid mosaic model is the presence of cholesterol. Cholesterol reduces the fluidity of the membrane and also works in reducing further escape or entry of polar molecules through the membrane. The fluidity of a cell membrane is mainly influenced by two factors: the amount of cholesterol that it has and the nature of the fatty acid, whether it is saturated or unsaturated. Okay, students, let's do the following questions from the information that you gain from the presentation. Question number 1. What is the role of carrier proteins in membrane? What is the role of carrier proteins in membrane? Okay, students, if we say carrier proteins are integral proteins that are used to transport large particles through the membrane, whether by facilitated diffusion or active transport, you are right. Question number 2. What is the disadvantage of cholesterol to man? What is the disadvantage of cholesterol to man? Okay, students, the disadvantage of cholesterol is if we take it in an excess amount, it becomes deposited in the blood vessel. So it narrows the lumen, then finally leads for heart disease and stroke. Even a complication that we call it atherosclerosis that leads for hypertension. All of you know, hypertension is a silent killer. Okay, students, let's wind up our today's session. There are two types of cells, prokaryotic cells and eukaryotic cells. Based on endocyne binary theory, eukaryotic cells are evolved from prokaryotic cells. Fluid mosaic model is the acceptable model that we use to represent the structure of plasma membrane. Fluid mosaic models have several key features. This is all about our today's lesson. Next time, we will see on how substance crosses the plasma membrane. Till next week, bye-bye.